Diandra, how are you doing?
I'm good, how are you?
I'm great, thanks for asking. Thank you for doing this interview. So let's get started. Alright, so the first question is, what are you working on right now?
So I'm working with Listeria Monocytogenes, and that's a gram-positive facultative intracellular pathogen. And it can survive in various environments, including the water, soil, and food processing plants. So people mostly hear about Listeria when during recalls, where like you have some contamination in processed meats, such as deli meats, hot dogs, or in dairy products and ice cream, as well as in fruits and veggies. So my lab is like a bacteria pathogenesis heavy lab, and so we do a lot of genetic manipulation. So what I'm specifically working on is a pheromone that is secreted in Listeria that enhances bacterial escape.
Well, what's the significance of your research?
So this pheromone is an example of diffusion sensing, and so it's a little different from quorum sensing, where if you don't know, quorum sensing involves cell-to-cell communication. So diffusion sensing involves one cell communicating with itself. And so this is something that is new and innovative with bacterial peptide pheromones. And also, I've recently found that complete loss of this pheromone alters viability of Listeria.
So what kind of techniques do you use in your work?
So I mostly use phage transduction that involves the transfer of genetic material via using bacteriophages. So the way that this works is that a bacteriophage injects its own phage DNA into a donor bacterium. And then phage proteins are made and nucleases are made that breaks down the donor bacterium's chromosome. The phage begins to repackage up its own phage DNA, but every so often it would package up some of the donor bacterium DNA. And this donor bacterium DNA have some type of antibiotic resistant cassette within its chromosome. So the phage particles are collected and then used to transduce into a recipient bacterium where recombination occurs, and then you can select on the transduced bacterium based on the transfer of antibiotic resistance that was present in the donor bacterium. So I do phage transduction in order to move the pheromone deletion mutant into other Listeria backgrounds. And so I also do some cloning techniques where I use PCR and vectors and plasmids to make mutants, since our lab is very genetic heavy. And I also do some cell culture techniques to test these mutants in in vitro assays.
So what's your career goal?
So I want to go into academia, or to stay in academia. I plan to do a postdoc when I'm done with grad school, and then I want to become a faculty member that has my own research lab. And so I thought about this a lot because I did want to go into industry, but right now I noticed that there are not a lot of African Americans or African American women in as faculty positions in higher research institutions. And so these institutions are mostly predominantly white. So when you have these African American students, they really don't have someone to look up to that's in their field. And so I want to be that person to go in and have my lab and have a robust career.
So what do you like doing when you're not in the lab?
So outside of lab, I spend a lot of time with my family and friends. I'm married, so I do lots of things with my husband, and we also have 2 puppies that takes up a lot of our time. So I'll go out with some friends or like to have dinner and just enjoy life, because lab can take a lot of your time. So it's nice to have a mental break.
So what kind of advice do you have for aspiring graduate students or current graduate students?
So I would advise that you work in a lab during undergrad, or at least do a post-bac, because this is a commitment. So you have to really know that working on your PhD is something that you want to do. It's very time consuming, and to not give up very easily, because this is hard. It is extremely hard, and so you have to be motivated and to continue working.
All right, thank you so much for this interview. Your work is amazing, and by the way, happy Black History Month.
It was great to do this interview. See you later.